Hey there, welcome to our channel, where we break down complex biochemistry topics into bite-sized, easy-to-understand chunks. Today, we're going to talk about homocysteine metabolism and its associated disorders. This is a crucial topic for medical students studying biochemistry, so let's dive right in. So, what is homocysteine? It's a non-essential amino acid that's produced as a byproduct of methionine metabolism. Homocysteine is then converted into cysteine or methionine through two main pathways: the transulfuration pathway and the remethylation pathway. Now, let's talk about how homocysteine is generated in the body. Methionine adenylyl transferase, MAT, catalyzes the reaction between methionine and ATP to form S-Adenosylmethionine or SAM, a very important methylating agent in the body. When S-Adenosylmethionine methylates an acceptor, this leads to the production of an acetylated acceptor and the production of S-adenosylhomocysteine, as indicated on the slide. An example is norepinephrine being methylated to form epinephrine. S-adenosylhomocysteine is subsequently hydrolyzed to form homocysteine and adenosine, as indicated on the screen. Two things could happen to homocysteine, as would be explained in the next slide. Homocysteine can be methylated to form methionine. This reaction requires vitamin B12, methylcobalamin, and N5-methyltetrahydrofolate, a mobile coenzyme form of folic acid, as coenzymes. The reaction is catalyzed by an enzyme known as methionine synthase. The enzyme can also be called homocysteine methyltransferase. A very important point to note is that both vitamin B12 and folic acid are essential for this enzymatic reaction. The alternative thing that could happen to homocysteine will be discussed in the next slide. Homocysteine can undergo an alternative path, known as the transulfuration pathway. Here, homocysteine combines with serine to form cystathionine. The enzyme that catalyzes this reaction is known as cystathionine beta-synthase, or just cystathionine synthase. This enzyme requires pyridoxal phosphate, the coenzyme form of vitamin B6, as a coenzyme. Thus, vitamin B6 is essential for this reaction step. Cystathionine is subsequently hydrolyzed to generate cysteine and alpha-ketobutyrate. The reaction is catalyzed by cystathionine lyase, which is also known as cystathionase. The enzyme also uses pyridoxal phosphate as coenzyme. Thus, cysteine can be generated from methionine in the body and is therefore a nutritionally non-essential amino acid. Alpha-ketobutyrate is subsequently broken down in the body to form propionyl-CoA. Thus, methionine is one of the amino acids that can give rise to propionyl-CoA. Others include isoleucine, valine and threonine. Propionyl-CoA metabolism is a very important pathway in biochemistry, but we are not discussing that in this video. Now, let's talk about the disorders associated with disruptions in homocysteine metabolism. These include homocystinuria, a genetic disorder characterized by elevated homocysteine levels in the blood and urine. We also have hyperhomocystinemia. These disorders are linked to an increased risk of cardiovascular disease, stroke, and dementia. Hyperhomocystinemia is a medical condition characterized by abnormally high levels of homocysteine in the blood, usually above 15 μmol per liter. This condition can be a risk factor for various diseases, including cardiovascular disease. It can be caused by genetic disorders, kidney disease, alcohol abuse, or poor dietary habits. For instance, deficiency of vitamins B12, B6 and folic acid can cause it. Homocystinuria, on the other hand, refers to a genetic condition characterized by high homocysteine levels in blood and urine, caused by enzyme deficiencies, especially the cystathionine beta-synthase. Therefore, hyperhomocystinemia can be a feature of homocystinuria. Features of homocystinuria are displayed on the screen. You can have a closer look at them. One of them is the displacement of the eye lens, a condition known as ectopia lentis. Enzyme deficiencies that can cause homocystinuria include the following: 1. Cystathionine beta-synthase. 2. Methionine synthase, homocysteine methyltransferase. 3. Methylene tetrahydrofolate reductase, MTHFR. We have talked about the reactions catalyzed by cystathionine beta-synthase and methionine synthase earlier. Now, let's talk about methylene tetrahydrofolate reductase, MTHFR. This enzyme catalyzes reactions that involve the conversion of N5, N10 methylene tetrahydrofolate to N5 methyl tetrahydrofolate. This is an essential step in 1-carbon metabolism. Without this reaction, there would be no N5 methyl tetrahydrofolate, which is supposed to react with homocysteine to form methionine. Deficiency in any of these three enzymes would therefore lead to the elevation of homocysteine levels in the body. So, how do we diagnose and manage these disorders? We use a combination of blood tests, such as plasma homocysteine levels, and genetic screening. We also look for clinical signs and symptoms, such as cardiovascular disease, neurological disorders, and eye problems. These are the key takeaways from today's video. 1. Homocysteine, a derived amino acid, is produced during the metabolism of methionine, an essential standard amino acid. 2. Homocysteine can be remethylated to form methionine. That reaction is catalyzed by methionine synthase, which requires vitamin B12 and folic acid for action. 3. Alternatively, methionine can continue with the transulfuration pathway and form cystathionine, which will subsequently break down to form cysteine and alpha-ketobutyrate. Both cystathionine beta-synthase and cystathionine lyase required for these reactions use pyridoxal phosphate as coenzyme. Hence, vitamin B6 is essential for those reactions. 3. Hyperhomocystinemia, which means elevated homocysteine levels in blood, can be caused by genetic factors, poor nutrition, alcohol abuse, or health disorders like kidney failure. Hyperhomocystinemia can cause several health problems, including cardiovascular, neurological, and eye problems. 4. Homocystinuria is a condition where there is elevated homocysteine in both blood and urine and is usually caused by genetic reasons. Possible causes are the deficiency of homocysteine beta-synthase, methionine synthase, or methylene tetrahydrofolate reductase. Associated disorders include stroke, cardiovascular disorders, and ectopia lentis. Hence, homocystinuria is a contributor to hyperhomocystinemia with its attendant problems. 5. Proper diagnosis and management of hyperhomocystinemia, homocystinuria are very essential in order to have good outcome. 6. Folic acid, vitamin B12 and vitamin B6 can lower homocysteine levels in the body. Therefore, an adequate diet that contains these vitamins is essential. These vitamins could also be taken as supplements to manage the condition. 7. Elevated homocysteine levels are associated with low levels of methionine and S-Adenosylmethionine. There are some important methylations of DNA and proteins that are crucial in the developing neural tube during the first trimester of pregnancy. Deficiency of S-adenosylhomocysteine would make these methylations to be inadequate, leading to neural tube defects such as spina bifida and anencephaly. Administration of vitamin B12 and folic acid have remarkably reduced the incidence of neural tube defects. In summary, homocysteine metabolism is a critical process that's closely linked to our overall health. As medical students, it's essential to understand the metabolic pathways, associated disorders, and clinical significance of homocysteine metabolism. Thanks for watching, and we'll see you in the next video. If you have any questions or topics you'd like us to cover, please let us know in the comments below. Don't forget to like and subscribe for more biochemistry content.